Hello, everyone. My name is Shreshth. Very excited to be here today to talk a bit about some of the work we've been doing at Deep Genomics, how we've used deep learning for a practical application in RNA biology, and also talk a bit about how we sort of tackle the prevailing notion around deep learning models not being interpretable, and we sort of managed to build a system that can not only produce a prediction, but also produce a hypothesis that you can test in a lab. Um, so I would like to introduce you first to the problem of RNA biology and how it's sort of uniquely suited to framing as a machine learning problem. We're all sort of familiar in the past few years with RNA vaccines, and the nature of RNA biology sort of uniquely lends itself to being viewed as a digital problem, and framing as a sort of machine learning problem that you can use that you can use data-driven approaches to develop predictors for. The sequence of letters that you see here on the screen, that is an actual drug. So by constraining your problem to RNA therapeutics, you can reduce your search. You can reduce the drug discovery problem to really a search problem of finding the right sequence of letters which achieves the desired outcome. And there's really three components that make this problem uniquely suited to AI. The first is really the component which is the modality of drug can be represented as a sort of piece of digital information, which is very uncommon in other forms of drugs like small molecules, where all you need is a unique sequence of letters, which is A, U, G, C, which computers can really easily interpret. And you also have like a large amount of data probing very specific properties of RNA. And there's been a sort of large growth in these types of data sets due to the advent of next generation sequencing in the past decade. And there's really terabytes of data for specific properties of RNA that you can train individual predictors for. The third thing is, which is probably the most important one, is you can, by using the power of deep learning, where you're like really extracting meaningful features directly from the raw information, you're showing the model the same thing that the cellular machinery sees, which is just the raw DNA sequence. And by constraining your problem to only look at the raw DNA sequence, you can derive generalizable and causal rules and actually come up with interventions, not only explanations. So just repeating the point here with an illustration of what we call the central dogma of molecular biology, where the DNA sequence is really, it goes through these several steps before it's transcribed into protein, which has function. The first step is the DNA is sort of transcribed into mRNA, into pre-messenger RNA, and it undergoes this process of splicing in which certain parts of the sequence is removed to construct the sort of source code, which is your messenger RNA, which is then translated into a protein. And the example which I'm going to show here is really going to focus on this problem, where like you have your initial sequence of letters, which is just composed of A, C, G, and T, A, C, G, and U if we're talking about RNA. And how that initial code is then translated into a final set of instructions, which codes for your protein. And another unique innovation, which is related to RNA therapeutics, is this modality of drug called steric blocking oligonucleotides, where you can actually have this drug, which is illustrated there, which what it does is basically acts as a patch on your genome, and you can make it unique to bind to like a very specific location on your DNA, and intervene in any of the processes which are disrupted. So if you have, let's say, a change in your DNA which is causing a misregulation of any pathway, you can come up with a steric blocking oligonucleotide to actually reverse the effect of the misregulation. And I'm going to talk about a specific example of a disease where we applied this to. So Wilson disease is a copper metabolism disorder, which affects about 20,000 individuals in North America and Europe. And what happens in Wilson disease is you have loss of function of the ATP7B gene. And this particular disease, people had known due to like co-segregation and family analysis that the root cause of the disease can be narrowed down to this specific gene, but people weren't able to figure out anything further from that to actually come up with a therapeutic. So the variant was reported in 1997, but its actual mechanism was not understood. And what we were able to do here by building a predictor that like really accurately understands splicing is to actually come up with a hypothesis for what this single change in DNA, which is this T to G change in the in a specific location in the ATP7B gene, what does it exactly do? And we were able to predict with high confidence that like this mutation actually changes the splicing code in a way where you have a portion of the gene not being included in the final transcript, and that leads to a protein which doesn't actually work the way it should. So just to introduce how we frame this problem as a machine learning problem, we have your input DNA, and this is like 6 billion letters. Each of the genes is transcribed into a pre-mRNA. And we also have examples through RNA sequencing of which parts of the initial sequence are actually included in the final mature mRNA. So you can imagine this is like a segmentation problem where you have an input DNA, and what you're trying to predict is like which parts of the input are removed from the initial pre-mRNA and are included in the final mature mRNA. So this is where splicing occurs normally. This process happens, millions of times every day in all of our cells. This is splicing is a sort of core part of gene expression in eukaryotes, and almost 95% of our genes are alternatively spliced. So what happens when you have changes in your genome, which are either inherited or you have a de novo mutation you have a germline mutation occurring at the time of fertilization, then you can have these genetic defects where like a single change can cause misregulation in splicing, which in this example causes the middle part of the sequence to not be actually included in the final mature mRNA. You have a shorter protein being produced, and it doesn't have the same functional properties as the full length protein, and you have loss of function. The way we sort of model this problem is to actually use a dilated convolutional network inspired by temporal convolutions, and we took the entire input pre-mRNA and predicted the final spliced product. We used several outputs for this network, one being the actual binary track, which is like the final mask that the cell sees of which parts of the sequence are included in the final splice transcript. We also added what we illustrated here as the green and red parts of what is the beginning of a sequence of included sequence and the end of sequence. What makes this problem slightly more challenging is that for most natural transcripts, you don't really have a single correct answer of a single final spliced outcome. Most genes undergo alternative splicing. So for the same input pre-mRNA, you can have multiple different outputs. So we had to encode that into the green and red track of all possible start points and end points for splicing to occur. We encoded them and encoded their rate of inclusion in the final transcript. And by doing this, we were able to train a predictor where this is actually showing you the real output from the predictor, where we take in the pre-mRNA sequence. Here, we're not actually showing you the letters here because what the input here spans over 20,000 characters. And the use of dilated convolutions here is important because we actually want the model to be sensitive to long-range dependencies. Because many of the introns are sometimes greater than 20,000 base pairs as well. And we also wanted the model to be very sensitive to small changes. So in the Wilson disease example, when we actually changed just a single character in this 20,000 character string, we were able to see very clearly that the model predicts that this middle part, which was previously included, will no longer be included. And this was able to sort of very clearly illustrate to us what actually happens when you have this like single character change. You have misregulation of splicing, and this leads to the middle exon, which is exon 6 in this case for the ATP7B gene, to not be included in the final product. So this was a hypothesis we could test with an experiment, and we did exactly that. So we constructed a mini gene system, which is just in a synthetic experimental construct. We introduced the mutation. We did another experiment where we actually took a cell line and edited in the variant with CRISPR, Cas9. I'm not showing those results here, but they confirm the same result, where we saw that by inclusion of this mutation, you had very clear signal showing that this mutation actually causes the middle exon, the one marked in blue there, to not be included in the final transcript. And by pinpointing the exact mechanism to investigate, you could have, for every individual, there's like thousands of mutations, and even in databases like ClinVar, which aggregate known mutations for disorders, there's over 250,000 mutations where people don't actually know what the mechanism is. So for, even if you knew the gene that you're interested in, there's almost too many mutations where you can, you would have to investigate all of them individually and actually come up with an experiment to confirm the hypothesized mechanism. But by sort of pinpointing the exact mutation we should look at and the experiment you should do to confirm the mechanism, we were able to establish the exact mechanism in a matter of months. What we started from was a list of 250,000 possible mutations in ClinVar. We were looking for mutations which were where the mechanism was not known and we knew would probably operate by altering splicing, and this was a very clear example which demonstrated that. What happened next was even more interesting, where not only were we able to actually say that this is the mechanism for the disorder, we were actually able to program a therapy for remediating its effect. So I talked a bit about steric blocking oligonucleotides, which is a form of RNA therapy. And we were able to, by using the same predictor and by perturbing its inputs and looking at its gradients, we were able to actually figure out what can you actually change in the mutated gene, which parts would you effectively mask out to actually restore the inclusion of the exon back to normal levels. And by generating those scores, we were able to, within a small experiment of just looking at around 50 to 100 compounds, we were able to actually find compounds which restored the effect back to normal levels. And this is, the sort of key thing to notice here is that the model had actually learned something causal about biology, because not only can you predict what the mechanism is, you can actually come up with an intervention to reverse its effect. And this is sort of very uncommon for models of biology. Even models like AlphaFold, they struggle at actually predicting, they do very well at predicting protein structure, but if you want to actually see how the effect of mutations are for the misfolding of proteins, they actually don't do very well. So it's difficult to train a model which relies only on sequence. For instance, AlphaFold not only uses protein sequence, but it also uses multiple sequence alignment, which is like additional evolutionary information that you have to feed into the model to actually get good performance. But because you're adding in that extra non-causal information, the model can't actually generalize to changes or interventions. But by doing that, in this case, we were able to actually find a compound which remediated the effect of the disorder. So just talking a bit about like our whole philosophy with using deep learning for these problems is changing the way that the traditional pipeline for drug development works, where you actually have all of these like waterfall steps of coming up with a variant, coming up with the mechanism of the variant, looking at the therapy, what the actual therapy does. All of these steps carry a risk, which are sort of unique to every new program or new gene or new therapy that you want to come up with. There isn't really a central component which drives all of them. And what we're sort of endeavoring to do is build this like shared understanding in the middle through these models of RNA regulation, which let us sort of generate predictions at all of these steps and let us sort of forecast into the future. So another thing we've done is not only do we have models for human splicing, but we also have models for mouse splicing and non-human primates. So it also lets us know if you were then to actually test this compound in animal screens, if you anticipate the same results later on. And that's really important for drug development because the most expensive failures are the ones that happen at the very late stages. So by predicting all throughout the process at the beginning, we're able to actually de-risk therapies by a significant amount. That's what we're sort of working towards. So I already showed this example of given a mutation or a variant that acts on the RNA, we can actually predict the variant effect. I showed the example of splicing, but we have several RNA regulation predictors that do the same thing, but for different processes. You can predict the mechanism of action for the variant. You can also then come up with the therapy, which is an SBO, steric blocking oligonucleotide. We can actually predict the therapeutic effect of it. And there's also various other processes that we can predict, like the safety and toxicity of compounds, coming up with intelligent biomarkers, identifying patient populations where therapy is expected to be more effective versus not because they have different genetic makeups. And finally, just to summarize what we've talked about so far, is what deep learning allows us to do is to really not have to force domain knowledge into our models, which would allow them to be non-causal. But by extracting raw features from our signals, just using raw DNA as the input, we're able to sort of train generalizable and causal models which can predict testable hypotheses. We've shown that we can actually test these hypotheses in experiments and de-risk several of the guesswork-related aspects of drug development, where for most of our therapies, we actually don't know the mechanism by which they act. And by actually being able to, in certain classes of therapies, approach it with that framework of understanding from the beginning, it sort of leads to higher probability of success and a sort of clearer understanding of what we're going for. The images I'm showing here on the right are from mouse primary neurons. And what we're also building towards is not only using the predictors for RNA biology to actually understand mechanisms, but also forecast even further into the future, predict things like the biodistribution of our compounds and potential ways of failure for our compounds in the future where we actually dose animals or humans with them. And hopefully that gives people an idea of what we're trying to do. Our mission is to decode the entire process of genome biology to program life-changing medicines. We believe that for most disorders, not only for genetic diseases, where in the rare example that I showed previously, that in that example you have a single nucleotide change which causes the disorder, but for most disorders, there tends to be a genetic component to it. certain people with different genetic backgrounds are sort of more predisposed to certain disorders, or you can actually play with the gene expression by upregulating or downregulating genes to act on disorders when they have even a small amount of a genetic component. So our mission is eventually to build up this understanding of genome biology by encoding them in our predictors and being able to actually program medicines for any condition and any population. Thank you.